Hi, and welcome to Christina Explains Difficult Things by Drawing Them Hour. Just kidding, this won't take an hour. So, in my 232 class, there were some really good questions asked about a quiz question that was concerning number of receptive fields in a particular patch of skin in this example, and sensitivity versus resolution, and what the difference between those two is. It is a tricky concept to understand if you've never thought about it before, so I thought I would go ahead and do one of these Khan Academy style sort of draw it out videos to better illustrate the point. So away we go. All right, so let's get some colors going. So first, let's define sensitivity versus resolution. So I'm going to get a different color here. Okay, so let's sensitivity is defined as the smallest amount of difference in a quantity that will change the reading of an instrument, but the instrument we're talking about here is a receptor cell or a neuron. So let's redefine this in physiological terms and call it the smallest stimulus value that will that will result in a neural signal. So an insensitive neuron and a sensitive neuron are two different things. Both respond to stimuli, but each of them requires a different magnitude of stimulus to be excited. So let's first give the example of a high sensitivity neuron. So for a high sensitivity neuron, a very small stimulus will result in excitation of a neuron. So we're going to say is capable of being detected. In a low sensitivity, oops, there's an N in there. I was writing faster or thinking faster than I can write. In a low sensitivity neuron, a large stimulus intensity is required to excite that neuron. Or the receptor, as the case may be. So you can have sensitivity on the level of the neuron, and for a sensitivity difference in an individual neuron, that could look like having more or fewer ion channels that respond to a particular thing. So a high sensitivity neuron might have many ion channels. So let's say that the neuron is acid sensing. So an acid sensing neuron has channels that are going to open when a proton is detected. So open in presence of H+. So let's say that neuron A is high sensitivity. So it has 12,000 H+ sensitive channels per square micron of membrane. So number of receptors is correlated with the sensitivity of a neuron, and 12,000 per square micron is a lot of channels. So this neuron is very sensitive to acid. A low sensitivity neuron It may be capable of being stimulated by acid, but it only has about 10 H plus sensitive channels per micron of membrane. So if this neuron is sitting in the extracellular fluid, as they do, and let's say 4 protons come along in solution, neuron A is much likelier to bind and detect those protons, even though there's only 4 of them, than neuron B. So that's what sensitivity looks like on the scale of the individual neuron. But we also have to talk about receptive fields. So a receptive field is made of more than one neuron in an array. And the size of the receptive field, the number of receptive fields in a given surface area, and whether or not those fields overlap is going to determine how sensitive the system is in that spot and how high the resolution is in that spot as well. So I'm going to erase all this and we're going to draw that out next. So let's draw ourselves a couple different patches of skin. Let's use skin for this example just for simplicity's sake. So we're going to draw in some patches of skin and I'm going to make them be parallelograms. So here is skin patch A. Here is skin patch B. Actually, I don't like the size of this one. I want them to be roughly equal sizes. So I'm going to modify my skin patch B to resemble A a little bit more closely because we want them to be comparable to each other. So let's fix that. All right, now they're of similar size. So let's go pick another color for my neurons. And I'm going to choose, let's do blue. Okay, so blue equals nerve ending. So it doesn't matter where the nerves go. I'm just going to draw the areas of the skin patches that they innervate. So let's start out by having skin patch A has one nerve ending. And the receptive field for this nerve ending I'm going to outline in a dashed line. And skin patch 2 has 2 nerve endings, and their receptive fields I will also outline. So when we talk about resolution and sensitivity, we just talked about sensitivity on the level of a neuron and saw what that looks like, but what about in a patch? Well, sensitivity and resolution is often best illustrated in a relative fashion, so comparing one thing to another. So that's what we're going to do here. So if we know that sensitivity means how sensitive a particular sensory array is to a stimulus, and we compare system A here with system B, which one is more sensitive? Well, B is more sensitive because it has 2 sensors instead of just 1. So B is more sensitive and A is less sensitive. Okay, that's all well and good. Now notice these 2 receptive fields are not overlapping, meaning that this system also has better resolution. So deflecting the skin surface here and here at the same time are going to result in 2 neurons lighting up. And so skin patch B will be able to detect that 2 points on the surface are being poked instead of just 1. So going back to our skin patch color, let's draw some more skin patches. So I'm going to draw C and D. Okay, so now you have skin patches C and D, and let's look at some different scenarios with multiple neurons that can change sensitivity, and can also change resolution. So just give me a minute to draw, and then I will explain. So what I'm drawing here is a sensory array with 1, 2, 3, 4, 5, 6, 7, 8 nerve endings in this skin, so already quite a bit more sensitive than either A or B because more areas that are possible to excite by touching. And also notice that the sensory receptive fields of these aren't overlapping. So if I get some calipers and I put them at some distance apart, and I poke the skin here and also here, because these receptive fields are not overlapping, my brain is able to detect that 2 points on the skin surface are being stimulated. So on skin patch D, let's illustrate a scenario in which there are the same number of receptors, but the receptive fields overlap. So I'm drawing in my dendrites. So we have 3, 4, 5, 6, so I need 2 more. And let's give those an attachment to some axons, because these are unipolar cells, so the cell body is going to be off in the dorsal root ganglia, not anywhere near the sensory array. Okay, so I have the same number of nerve endings, 1, 2, 3, 4, 5, 6, 7, 8. But now, let's draw their receptive fields. I'm actually going to pick a different color for this just to illustrate my point a little bit more clearly. So now I'm going to outline the receptive fields for these in red. So the receptive field for this one is here. The receptive field for this one is here. And just for comparison's sake, let's do the same thing on skin patch C. So oops, we don't want them to overlap, so undo. There we go. They may be closer, they may touch, but they shouldn't overlap. Okay, so we have here non-overlapping receptive fields, and here we have overlapping. So let's use the calipers example again. So let's say I put the calipers here and here. Because this particular caliper point is overlapping, or in the zone of overlap between this receptor and this receptor, both of these are going to fire action potentials in response to that stimulus. Same deal here, so this one and this one. So you have created a situation where at the same sensitivity, so same number of receptive fields, you have lower resolution. Resolving power is the ability to discriminate between one thing and another, so to tell two discrete things apart. From the perspective of the central nervous system, the nervous system knows that these lines are labeled for these particular skin locations due to somatotopy or mapping, but from the brain's perspective, even though we can see that the caliper is in this particular point, because these two neurons are being excited, and these two are, what the brain thinks the receptive field is, or thinks that the point being stimulated is the following. I'll outline it in orange. So from the brain's perspective, it only knows that the area bounded by the orange is being stimulated, not which particular area within this patch of skin. And same deal here. So I'm going to color that in. There we go. So the orange is where the CNS thinks the touch is happening. Oops, I ran out of room. So that's the CNS interpretation of touch location, even though in actuality only the point being stimulated by the caliper directly is really being touched. So you lose resolution and you lose discriminating power by having receptive fields overlap. So let's talk some more about the quiz question, and I will explain now that we have fleshed out these topics. Okay, so I went and got a hold of the question that I asked in lecture, and oh man, I must have had morning brain on because I see a problem with it, and I will explain. So let's just write the question first. It was question 4 from the quiz, and it said, fill in the blank. The more receptive fields a sense organ has, the better its something will be. Okay, so the options were as follows. A, sensitivity. B, resolution. C, accuracy, whatever that means, obviously not the right answer. And what was D? Ah, yes. D, all of these. So I said the answer was resolution because I was having a dumb moment. So it could be resolution, but it also could be sensitivity. And I think that's where the student that was asking those questions was getting confused. And I totally see why now that I am fully caffeinated and thinking the problem through. So I apologize. Part of being a good scientist and a good teacher is admitting when you are wrong. So this is me admitting that I was wrong. I am sorry. Let's make it better. Also BT dubs. For that reason, everybody will get this point. So Let's game this out now that I've identified the problem, and we can talk about why the answer could be resolution, but it also could be sensitivity. So, more receptive fields could equal more sensitivity, but this doesn't acknowledge channel number or sensitivity of the individual neurons. So this is also a candidate answer. Resolution is what I was thinking of because I wasn't paying attention to the wording of the question. So what I had in mind, what I saw in my brain when I made this question, was a receptive field that looked like this. This was my intention, but again, I could have worded it better. I also could have drawn a better rectangle. That rectangle sucks. All right, let's try that again. Better-ish. Okay, so floating in my brain during the authoring of this question was a receptive field that looked kind of like this. So I'm going to make lines instead of meticulously drawing circles. So in this picture, each square is a receptive field. And none of them overlap. So this particular sensory array has good resolution, and presumably because there are many receptive fields and therefore one neuron per field, good sensitivity as well. So that's why the answer could be either, now that I think of it. I hope that makes sense. So you could change this. It could have the same sensitivity but crappier resolution if all of these overlapped, because then the brain wouldn't be able to tell definitively, is square A being poked or square B being poked? I'm not sure. I'll just assume it's both. So you can cram lots of receptive fields into a small area and overlap them. If you really want to jack up sensitivity, but if you do that, and the operant term here is overlap, if you do this, it is at the cost of resolution. So that's the basics of the relationship between sensitivity and resolution. They are not the same thing. They are related in a trade-off style relationship with regard to sensory arrays in human and other animal physiology. So I hope that clears things up. I am going to go grade the quiz now, keeping this in mind. And I also want to thank you guys for asking such good, astute questions. It helps me be a better teacher and think more carefully about how to teach this stuff, because it really is tricky and I'm used to knowing it, so it just sticks in my brain just fine. And sometimes that causes me to forget what it's like to not know it in the first place. So working together to make both of us better. All right, I will see you for the next video. Thanks for your attention.